sub-teams. So Andre, who's going to speak in a moment, is leading our AI sub-team. And the aim there is to develop cutting-edge AI to enable multi-omics cancer biomarker analysis and AI-assisted clinical decision support. We have a technology solutions team run by Alex Bogaty. So this is the team where we have software developers who can build tools, systems analysts who can maintain our Azure platform for secure data, secure clinical data transfer. And we have a technology trials team. So Dr. Donna Graham, who's an oncologist at the Christie, is in our team 1 day a week, and she and her team have the capacity to run clinical trials of new and innovative technologies. So we're quite a diverse team in the skills that we bring. And this slide is done slightly crazy, but there's about 18 of us in the team now altogether, and with quite a wide range of skills. So we have the capacity to develop new algorithms, build those into digital tools, and run trials to implement them in real clinical settings. So we've got skills around clinical trials, patient engagement, software development, all kinds of different stuff. So it was a real gift for me to join the team towards the end of last year. Our current funding is largely through Cancer Research UK. We have a big CRUK accelerator grant called UpSmart, where we're designing digital tools and open sourcing them to support early phase clinical trials for cancer. And we're doing that in collaboration with groups in Italy and Spain. We've got a bit of Horizons funding, and also we're part of the Manchester BRC, so the big NHR Biomedical Research Consortium. And we have some core funding as part of the Cancer Research UK National Biomarker Centre as well. So as I mentioned, one of our key interests is around early phase clinical trials for cancer. So I thought I'd just spend a slide to talk about that a little bit, because it's a really interesting space. I don't know how many of you are kind of clinical trialsy people, but I thought I'd talk through the background to that. So the figure we've got here on the left of the slide is the kind of traditional view of how the drug discovery process goes for new cancer therapeutics. So what's 1, 2, 3, 4, 5 steps here, usually takes around about 10 years and costs somewhere in the order of $1 to $2 billion per drug. So this is an enormous process. Starting from preclinical studies, where you're kind of looking in lab models, in animal models of a new drug, and then moving into phase 1 studies, which are traditionally the kind of safety studies for new drug. So this is where a new drug will be tested in patients for the first time. Usually these are patients who have no other treatment options available to them. So they've tried all the other licensed treatments for their disease and nothing's working. So they're offered clinical trials of a new therapeutic. The key questions to be answered in these phase 1 studies used to be, how much of this new drug can a patient tolerate? So it was more about looking at the safety of a drug rather than the effectiveness. But over time, that's changed. So with the introduction of more targeted therapies for cancer, so therapies targeted against particular gene mutations, and also the immunotherapies that have come through in the last 10 years or so, those phase 1 trials can actually have much more benefit for patients. So instead of being just an extended lab experiment, these are now therapeutic options for a proportion of patients. There's some figures on of around, you can see figures for suggesting that 20 to 40% of people on phase 1 trials might now be getting some therapeutic benefit. That might be a bit exaggerated because of underreporting of trials that are not successful. But we can see that there is a definite improvement, that these are now genuine clinical options. So something we want to do is to be able to run those trials smarter. In phase 1 trials, we're still really interested in collecting toxicities data. So are patient's kidneys being damaged by a new treatment? Are their eyes being damaged? Is their heart damaged? How can we use new technologies, new algorithms to detect those toxicities better, to predict who might experience those, and to really gather and analyze data in a more effective way so that we can really improve that space? So that's where we are. And I think I was going to, so I'll talk through a few of the projects we've got ongoing. So if this is a very stylized kind of image of the processes that might happen in an early phase trial, across the kind of central arrow. And in the blue and grey boxes are some of the projects that we have running within our team. So we've got a number of digital products that are in development. So that could include some of the multi-omics biomarker analysis to support hypothesis generation. We've got tools to support clinical trial design like Treetops, which is about minimizing the environmental impact of a new clinical trial, or inclusivity guidelines about how to include a wider and more diverse range of people in early phase clinical trials. We're looking, clinical trial planning and approval processes are immense, unending bureaucratic exercises with just endless paperwork, nods from Andre, endless paperwork to be filled in and a lot of duplication of effort. So can we use large language models to make that process more efficient? We know that the government's really interested in making the UK a more kind of attractive space to run clinical trials because that's a real source of income nationally as well. So could we use large language models to speed up those processes to make it more efficient? So thinking about patient recruitment, we have a clinical trial matching tool that we've developed that can search the clinical trials registries, which are immense in the amount of data they include, but to do some intelligent searching on those to be able to match a patient to a clinical trial that might be suitable for them. And we've got a couple of tools, PIPO and ScreenFail, which are at different stages in development, but we're looking to try and predict who might be suitable to go onto a clinical trial at all. So who's well enough at that point of the first decision about whether to offer somebody a clinical trial, who's well enough to go onto those clinical trials and maybe find some benefit and offer us some informative research data. We've developed some clinical decision support tools. So eTarget is a kind of data visualization platform that's being used in molecular tumor board meetings in a big study called CRUK National now. So I think we've recruited about 2,000 patients onto that with a target of 6,000. So eTarget and it allows ctDNA data and histopathology data and clinical notes to be presented in one format and to allow decisions to be made more effectively in meetings rather than 10 different pieces of paper being shuffled around for each patient. Coronet was a tool that we developed during COVID to help with clinical decision making for COVID patients with cancer or cancer patients with COVID. And then in terms of patient and trial monitoring, we've got our QA tool that's under development, gathers information on clinical trial deviations. So when you set up a clinical trial, there are really fixed protocols about every single different step that a patient should have. So when should a test be taken? When should a treatment be offered? But often those plans aren't quite followed to the letter. And there are all kinds of different reasons why that won't happen. Like a patient is ill on a certain day, or a particular member of staff isn't available to do a certain thing on a certain day. But collecting that information across a trial, especially when that trial might be run in different sites, can be really informative because it could show patterns in how that trial is running that could have implications for patient safety. So the QA tool is looking to collect that data from across different sites within a trial to be able to present that data to the trialists, but also to the companies who are running those trials to make to again gather better information and use that in a better way. ProAct is a patient-reported outcomes tool, so it enables video messaging, text messaging and surveys between patients and their clinicians while they're on a study so that we can better capture who's experiencing different side effects. So, for example, somebody might have a headache one day but not think to mention it next time they see their clinician, but if they can keep contemporaneous notes and tell their clinicians day by day how they're feeling, then again, we're generating richer data that may be more informative. And for data visualization, we have our Acuity tool, which is a tool that was originally developed by AstraZeneca, but was handed over to us, but just to enable some visualizations of that complexity of data that is gathered during a clinical trial. We also have a few trials in progress. So I mentioned the CRUK National study already, which is using eTarget and our clinical trial matcher. We also have a few trials looking at in-home data collection for patients. So Notion is a study where we are, which one is this? So we are collecting data on nephrotoxicity. So patients are taking blood samples in their home and posting them into us so that we can monitor their immune-related adverse events from immune therapeutics for cancer. AI is a an artificial intelligence tool we're hoping to develop that can read retinal scans to detect whether somebody's experiencing retinal damage from the cancer therapeutics. And APACE is a study using a wearable device, a watch, that can measure patient activity and sleep, because we know that it can be difficult for clinicians to assess how well somebody is, in the sense of how able they are to go about their normal activities. So, which is important when considering whether somebody is suitable to go onto a clinical trial. So we're looking at whether using a wearable device is a better way of collecting that data. And the CC Work Package 12 trial is using our proactive patient-reported outcomes tool. So we have a lot of different projects ongoing within the team that are all around that clinical trial space. So hoping to collect better data and use that data in a more informative way to support clinical trials. So just to finish with a couple of acknowledgements before I hand over to Andre. So of course, we must acknowledge the patients who are letting us use their data in our studies, and our close associations with the Christie Hospital, the University of Manchester Research IT. So a couple of members of our team are based in Research IT and are kind of seconded to us. And the Experimental Cancer Medicine Centres Network, who we work with quite closely on our UpSmart grant. And of course, our funders. So unless anybody has any questions at that point, I'll hand over to Andre, who can talk through some of the projects in more detail.
Maybe we can do the questions at the end, unless you have any pressing questions. So thank you for the invitation. Great pleasure to be here. So Harriet showed you really the large portfolio of projects that we at the DCR work. So today, I set a slightly more technical angle, focusing on language models. So it's more of a computer science type of narrative. So the idea is not to detail everything that we do on that side, right? We have projects, for example, that are working with multi-omics types of data sets. I'm not going to discuss them, but we are going to focus on our strategy with regard to language models. So from the point of view of the research in NLP that we do, these are some of the keywords that maybe describe what we are aiming to achieve. So we are working at the interface between symbolic models and statistical machine learning models. We focus at the interface between reasoning in its more formal sense and inference in the statistical sense. Really, how to bridge these two worlds between the symbolic and the machine learning world. So this gives a little bit the theme of what we focus on that side. And as Harriet mentioned, a lot of this goes down into, or at least is highly motivated by biomedical applications, but I'm going to discuss some principles beyond that. So I'm part of the DCR team, but I also have an affiliation with the computer science department at the university and with an institute in Switzerland. So the whole set of contributors to some of the elements of this talk are expressed in this picture. So this is more or less the joint work of this team. So as I mentioned before, the goal, our research center or center of gravity is trying to develop models, in particular, natural language processing style models, which are capable of abstraction and reasoning, right? And this is important in the clinical context, but also important in the scientific discovery context, right? And essentially, we are motivated in the context of oncology to develop these models. So our emphasis, technically speaking, is natural language inference. So this is really our research expertise. And we work in terms of application areas in oncology, but also in mathematics, in physics. So everything that's motivated in terms of scientific discovery, scientific inference. So how to build language models that can do rigorous scientific reasoning. So technically speaking, a lot of our research is about improving the control of this model, these models. So really how to prevent hallucinations, for example, how to allow these models to do controlled, rigorous, stepwise reasoning in the scientific sense. And I hope with the example that I'll give, this will become slightly clearer. So let's start with the motivation in terms of scientific discovery, which communicates very well with what Harriet was mentioning in terms of the clinical trials process, right? So clinical trials are about experimental medicine. depending on where you are in the cancer research, you want to discover, for example, new biomarkers, which could provide a better stratification of patients, or you want to discover a new drug, or you want, for example, to prevent and predict the situations in which patients, for example, have toxicity effects, right? So an archetypal analytical workflow, right, and this is one archetypal analytical workflow, starts with, of course, trying to understand the state of the art, the literature, the data, which is already available, right? So which could be in form of databases, text, and so on. So we select the relevant background knowledge. We have new questions which are grounded on this background knowledge. We may translate that into an intervention, right? So the application of a certain treatment to a cohort of patients. We collect new data, right? That will inform our discovery process. Then we go back and we abstract out of this data, right? So we make some analysis. So we essentially select and abstract over this data, and hopefully we explain a new phenomenon, or we improve an explanation of a new phenomenon, and we enrich an existing model, which we may or not corroborate with previous evidence again, right? So this is a quite relatable analytical workflow. So I think the vision, the motivation for what we'll discuss today lies on the fact that with language models, we have the inference substrate that can really optimize this process. So I think with these types of universal machines, we are at a position to think to what extent we can augment and automate parts of this process, right? So, and these are some of the areas that we are operating towards this vision of trying to augment this process, right? And behind the scenes, using artificial intelligence methods, but heavily grounded on large language models for all these phases. And I'm going to exemplify some of them here. So, but what is the connection between them, right? So, independent of what type of scientific research one does, the end products are linguistic products, right? So you may do your analysis, you may work on your lab. At the end, you publish, you communicate, right? That's an example of end product, a statement in natural language. Could be a table, still a linguistic artifact, numerical, categorical, or could be a more formal equational type of abstract model, still a linguistic artifact. So the common denominator is with language model and scientific discovery is language and abstraction, right? So this is the medium in which we communicate and we persist the end products of any type of research. And that's how it links to language models. So for those of you, I don't know how many of you here, certainly Goran has been on this area for a long time, but transformers are the engine behind language models, right? And transformers emerged more or less in 2017. And since then, there was a rapid evolution of these models in the direction of providing more nuanced, more complex natural language understanding and natural language inference, right? So I try to establish here a kind of simplified timeline of this process, but I think what was surprising, certainly I think Goran can confirm or disconfirm that, is that I think people in NLP did not expect that this architecture would work so well, right? So certainly it was a breakthrough at that time, but it wasn't clear that this engine would run so well as you scale up data acquisition, as you provide on the top of that new alignment models. So it was really a surprise that by 2022, you had ChatGPT, which was such a functional and universal machine that emerged, right? And now we have the commoditization of these models. So we have open source models which are available. We have, since the last 2 years, variations of these models which are now working over omics data, right? But still, there is a gap between the omics type of reasoning that you can do with these models and the natural language types of inference, right? And these are some of the areas that we are working on, both on the omics side and the natural language side. But I'm going to emphasize today the natural language side. So technically speaking, I selected a couple of projects that we are doing in the context of the DCR that are directly related to oncology research. And I decided to split this talk into two main areas, right? So first, the use of large language models as a mechanism to organize and select evidence. So a lot of the reasoning that goes within a clinical trial goes on trying to relate to existing evidence, to existing data, and to existing publications. And then the second is, how do you represent this evidence within a linguistic space? Which methods you can use to essentially build a kind of controllable memory so that you can build AI models which can reason over these facts, right? So, and that's essentially the motivation. And of course, downstream of that, the intuition is that with that architecture, one can essentially deliver better reasoning models, augmented reasoning models for oncology, scientific discovery in oncology, but of course, relatable to other areas as well. There's nothing really specific to what I'm talking about today. That's too oncology specific. That's not transferable. So let's start with an oncology problem that we worked at the DCR, which was the fact that we wanted to start building predictive models to predict toxicity, right? So some of these treatments can be very toxic. One of the types of toxicities is called CRS, cytokine release syndrome, for the immunotherapies. But the problem in trying to build a predictive model in this case is that the cohorts of patients that you have access to are around the order of 15 patients, right? So you cannot really work under, let's say, the constraints of traditional statistical inference. So the question that we asked ourselves is, can we enrich these models with existing background knowledge? And focusing on that extraction of background knowledge, can we automate this process of building this knowledge base of parameters using language models? And that's what I'm going to focus here, right? So what we started doing, we essentially built a model, did an extraction. We selected from more or less 500 papers, around 17 highly relevant papers. And then by hand, we extracted tables and parameters. And this took us more or less 40 hours, right? Then we built a language model-based pipeline that allowed us to essentially drop this evidence extraction by 300 by orders of magnitudes. So it becomes 300 times more efficient. So this is an example of what I'm talking about concretely. So given that you have a paper, you build a pipeline that's able to select the sections that you would like to focus on, and then, of course, extract the parameters and values that you need to extract. And then what you have here on the right-hand side is more or less this real-time filling of the parameters, which are cytokine-related markers, right? So just to give you some concreteness on what we're trying to achieve. Given sets of papers, produce a table with some guarantees, safety guarantees on what one is extracting on that side. However, to do that, you don't just ask ChatGPT to do that, right? So, and I think the message here is that one needs to move beyond seeing language models as a conversational aspect or as a conversational agent. So the language model is one box in this pipeline. And to deliver this scenario, you need to complement this pipeline with other boxes. And they can be quite banal, engineering-based, but not trivial to implement, which is how do you extract the layout of a document? How do you extract tables from a document, for example? How do you select relevant sections? And all of the steps are important for the LLM to do its work, right? On top of that, how do you augment the vocabulary of the model as you have a lot of synonymic variations of this, right? With, for example, an external ontology or thesaurus, so that you can do your extraction. So the message here is the language model itself needs to be complemented by the composition of a pipeline, and language model is one component, maybe the most important component. However, to operate, it needs to have other components developed together with it.
Andre,
Yes, please.
can I ask the table columns, are they predefined, or you leave the language model to populate the column names, if you want, as they scan the literature?
So you give as an input your set of parameters that you're looking at, and this indirectly means the table. Yes. So
that's your defined, basically the target.
Yes.
Prompt.
Exactly. So one of the projects that we did was a platform essentially to try to coordinate these elements required to build expert agents. The way that we're conceptualizing this is that if you have an information extraction pipeline like this, you're building an expert agent for that pipeline. That's an open source project called Lunar. And this is more or less how it looks within Lunar, right? So you essentially have on the left-hand side a set of components that you can reuse, extract PDF, extract the table from PDF, so on and so forth. And then you drag and drop and parameterize these boxes so that in the end you can have your table, right? So that's an example of how it looks like in practice. And this gives you an idea on, of course, it's accelerated, but how efficient this can become when you have your predefined components and that you reuse these components when you're building your pipeline. So this essentially relies on a library of components. So this project was a project developed outside the DCR, right? It's more universal in shape. And then at the DCR, we are specializing this framework in the direction of becoming more oncology specific, right? So a part of the motivation that we have for this is trying to integrate with external knowledge bases, which are very prevalent in the biomedical area and in particular in oncology. So how can you reduce the barriers for integrating evidence from databases, from the literature, from PubMed, and other data sources, which are nicely curated evidence, so that, for example, you can enrich a set of genes, for example, that would, for example, explain the differences between responders and non-responders, right? So then we generated a variation of this framework that we call BioLunar. And BioLunar essentially, I'm not going to demonstrate that, but essentially are the same boxes, but now we have a library of components that allows you to query pathways, to do gene enrichment, right? Not only querying external evidence, but linking to external tools as well, a technique that we call tool forming. So within Lunar, what one can do as well is to do workflow compiling, right? So the abstraction behind this is a workflow, but can you automate that even more? So given, for example, that you want to do a workflow that, given two studies, extract the gender distribution between the two, or the target study, right? You can communicate that in natural language, right? It's a bit typed quite slowly. But then what you get in the end is that you have a workflow that's produced automatically, right? It goes from natural language to workflow. Of course, this is not going to generate a perfect workflow, but at least will shape up, based on the existing components, a hypothesis on what would be a workflow. So it's another step of automation. So the idea here is really to reduce the barriers between domain experts and essentially generative models, in order so that people can create these expert agents without necessarily needing to program, right? Or having this specific AI expertise. So we are applying these types of methods, and I'm not going to describe them in detail, as Harriet mentioned, to really try to optimize the clinical trials pipeline. So as Harriet mentioned, there is a severe cost associated with every phase of this process. In some of the areas that we are applying language models involves patient trial matching. So given that you have really a large set of clinical trials which are available for people, how do you do this fine-grained matchmaking, reducing setup time? How do you automate the bureaucracy behind sending forms, filling forms, and so on? Optimizing screening, prediction of toxicity events, which I mentioned, and understanding protocol deviation. So these are some use cases that I'm not going to detail them, but that we are approaching using the language model strategy. Now, to the second part, I try to emphasize that with language model, communicate the message that with language models, we can have this enrichment, this collection of evidence at scale, right? Via a kind of meta-analysis, as was demonstrated with the table part, or by querying, for example, external knowledge bases and reducing the barriers for that. But then assuming that you now have this rich knowledge base of evidence that you onboarded into your evidence base. So how do you interpret that? How do you use that for reasoning? And then we're getting a little bit more technical, let's say, on the natural language processing side, right? So a way to conceptualize a inference is to see that as a chain of claims and premises, right? So this is the structure of an explanation. So if I want to justify this claim according to previous evidence, I can run through a knowledge base of evidence, and then I can essentially bind the previous premises to the claim that we're looking into, right? So, and that symbol there essentially represents a type of explanatory entailment, right? And this is essentially an explanation for that fact based on existing facts that we know, right? So one question that we have on the NLP side is how do we model facts and their relationships, the interment relation in a controlled manner, right? Because the vision is how can we build models which can explain medicine? How can we build models which can reason the way that an expert would do, right, from a natural language processing perspective? So, but then when we're talking about medicine, the understanding of the concepts, the definitions tends to be quite complex, right? So we have very specialized terms. And then a variation of this question is how do we build models which are capable of understanding complex concepts? And this relates to the general problem that you have in scientific inference. The fact that you need to interpret very specialized sets of concepts and you need to relate back to definitions and do that in a rigorous manner. So the types of models that we are aiming to build, or the type of inference that we are focusing, is essentially how can we bind the facts in a controllable way, right? Given that you have sets of facts, and this relates to the notion or to the problem of hallucination. And related to that, we need to have models which are capable of understanding concepts in a rigorous manner, right? Looking to the necessary conditions for that, for the application of that concept, for example. And this puts us more or less in a logical mindset. And that relates to the symbolic element that I mentioned before. So one of the areas that we are exploring in natural language is how can we transfer a notion of that we have emerged essentially in computer vision of disentanglement, right? That you, for example, when you're building a computer vision model, you have some semantic factors which emerge, right? So you have factors that you can name. A certain face will have some notion of age, gender, glasses, mustaches, hairstyles, and so on, right? And computer vision models are able to disentangle and then control these factors, right? So we have latent models. Behind the latent models, we can align some semantic factors, and then we can take an image, for example, and say, age this image, or change the gender of this image, put a glass, and so on. So we are doing a similar type of control with facts, with sentence embeddings, going in the direction of how can we use this type of disentanglement mechanism to control inference in natural language. So given that you have a set of facts, such as the one that we discussed before, we'll have a language model, right? So model here, you can consider that to be a language model. And behind this language model, we have an underlying geometry, a latent space geometry, right? And this geometry will have some consistency properties. So the analogy here with computer vision is that if you want to build a more consistent latent space, you can use this entanglement as a mechanism for that. And then you can intervene in this architecture by essentially injecting symbolic controls. So again, relating that to scientific reasoning and cancer, essentially, if we need to apply language models in the context of scientific reasoning or clinical reasoning, we need to have strong control guarantees. How do we do that? So it is an example of how we can conceptualize, right? So given that we have two explanatory facts, and then using a simpler example here of very basic biology, we have the underlying embedding geometry within the latent space, and then we can traverse this latent space and interpolate on this continuous sentence space. So you can imagine a sentence space of facts, right? And if we do that without control, this is what we get, right? So we get a bit of a jumpy semantics as we move from point A to point B, right? We see that the verb is changing, right? And we see that there may be an inconsistency in the nature of the subject and the object arguments, for example. If we do an injection of semantic controls, and this is an example of using semantic control labels, so this is essentially a linguistic structure that we can inject into the model, but there are other elements, we can have better separation, better latent space geometry, and then you can see that the traversal is way smoother. So you don't have this jumpy element between the verbs. So we controlled the geometry of the space by injecting an extra linguistic prior within the model. How we do that, I'll leave it still abstract as a reference. And then by doing that, we can organize our space in a much more internally consistent way. And then we can have better separation between the semantics of the sentence, the content of the sentence, the content semantics, the formal semantics of the sentence. And this is important if you essentially would like to have your model inferring with more consistency and more inference guarantees. So essentially what we are doing is relating a geometrical notion, right, this vector space notion, with this notion of separability of clusters and the notion of disentanglement. So the fact that we have latent dimensions which are specialized to encode specific elements. And then we use operations, vector operations, to essentially investigate and probe the space. So we have plenty of contributions in that specific space. So methods that we can use to improve the geometry of the latent space of the language models. Typically, we do that using a variation autoencoder type of architecture, where we have a language model encoder, we have a variational bottleneck, and then we have a language model encoder, right? And then we have the sandwich, and then we have other interventions such as invertible layers that we put, and that gives this extra control properties, right? So these are some of the architecture strategies and modeling strategies that one can do that. But there are many other strategies such as encoding graph structures, so many priors that you can inject within this model. But again, the intent is the same, is to have a representation of these facts in a more controllable manner. I mentioned that we need to, as well, represent concepts in a controlled and principled way. Another method that we are using that's geometrically inspired as well is based on hyperbolic embedding, right? So behind the notion of concepts we have or defining things we have, the notion of a taxonomy, we go from more abstract to more specific phenomena, right? And hyperbolic spaces capture this characteristic where at the boundary of the space, you have way more granularity, and at the center of the space, you have distances which are less granular, right? So by essentially encoding concepts in that controllable manner, we are able as well to have language models which conceptualize better and can use these conceptualizations for downstream inference, right? And this is how we encode domain-specific concepts. So now going back to something that we're starting to work, but this is kind of still early work, but it's based on the work of the group of Caroline Pula. So it's found that variational autoencoder types of architectures can be used for example, encoding and representing gene and tumor states, right? So instead, I was talking about autoencoding a sentence, right? A fact, input fact, regenerate fact. But you can think about having an RNA-seq type of input and trying to reproduce that RNA-seq. And with that, one can use the same control mechanisms essentially to try to model the state of a tumor, right? Given that you have, for example, the multi-omics types of inputs, can be other types of inputs, not only RNA-seq, but then trying to use this language ability of transformers, this expressivity of transformers to encode the tumor state. This is something that we're starting to experiment with as well. But of course, the extent in which these models are going to operate on the multi-omic space, there will be a gap because the access to the data when compared to natural language processing is significantly reduced, right? And then the idea is that we can, with that type of model, find better clusters of patients, do better patient stratification, predict, for example, directionally the intervention of a drug, right? So we move the tumor state from A to B, and this is essentially very analogous to what we discussed in natural language. But of course, the challenge is we're operating at an evidence base that's significantly lower. And of course, one needs to account for things such as batch effects and so on within that architecture. But there are methods, well-known methods for that. So maybe to finalize this element of control, what I'd like to mention is that one can as well bind and integrate different modalities, right? So some of the work that we're doing as well is about disentangling the structures of graphs, molecular graphs. For example, exploring architectural elements that have the symmetry-induced priors. And again, having better disentanglement, better generative control, but now for molecules. So then the assumption here is, can we put and align these molecular representations or the omics representations of a state with the natural language representation, right? So that we align all background knowledge within that reasoning space. So we have produced a library called LangBE, and this is a library that allows you to compose different types of architectures in terms of encoders and decoders with different types of bottlenecks for the VAE, right? So the idea is really to reduce the barriers for experimenting with these types of VAE style controls so that you can do the probes that you need to visualize the space, understand the properties of the space, and this is something that you can access as well as an open source tool. Now, in this last part, I'll just finalize with some comments on the notion of inference. So we talked about representing facts and controlling the nature of the facts in the latent space. Now, when we are talking about inference, we can come back to the notion of an explanatory inference, right? Given that we have a claim and a set of premises and facts that we consider to be true in a fact bank, how do we link that in a logical, so to say, manner? Logical, because this is not just a formal inference, right? Explanatory inference is very material content based, right? So how can we produce a type of explanation that's constrained and still use the content flexibility of language models? So this is some of the problems that we're looking at. And we have essentially experimented with some neurosymbolic methods. So one way that you can conceptualize this is, given that you have your knowledge base of facts and a certain conclusion that could be a question or a hypothesis, how do you bridge this gap between the evidence and essentially generate a proof or an argument that connects the basic facts which were selected to the conclusion, right? And select the facts which are relevant for that question as well. So this is a quite convoluted architecture. So, but just capturing more or less the spirit and the shape of the architecture. So this is a work that focuses on building end-to-end abductive solver. So abductive means essentially this explanatory inference, right? So that you're going back to the facts and explaining something based on the facts that you think to be true. So we start again with knowledge base of embedded facts, right? So these are sentences. We have the embeddings of the sentence, and then we do a retrieval process that will have a certain relevance function. And then in this middle part, using differentiable convex optimization, what we do is essentially we learn to link the facts, but using some linguistic and inference priors, which I'll leave it vaguely at this point. But then essentially we have an architecture that allows this end-to-end learning of how to train an explanatory model, right? And that's the spirit of that type of neurosymbolic intervention. Another type of architecture that we are exploring as well is the dialogue between large language models and external solvers, right? So you have symbolic solvers such as theorem provers, such as Isabelle, Prolog. You have equational solvers such as SymPy. So how do we put the language model to dialogue with something that requires this rigorous, stepwise, and precise understanding of formality? And essentially, the nature of the problem is you start with your facts in natural language, you auto-formalize these facts using the LLM into the language of the solver. this may run into a certain set of problems when you're trying to run that in the solver. You use the output and the log of the solver to refine your representation, send back to the solver, and then you iterate until you reach a convergence point, until a certain point. And this allows you to do this neurosymbolic dialogue under this two-former framework. And two-former essentially means this ability of the LLM to dialogue with external tools that are capable of, let's say, different types of reasoning that the LLM is not designed for, right? Could be as simple as a calculator, but could be, as we are doing here, something that's more logically symbolic based, right? And then at the end, what you expect out of this iteration is that you have your prediction, if that's the case, but also you have an explanatory tree that describes in a consistent way how these steps were achieved, right? So we have better stepwise inference, more completeness in terms of the facts which are required, and more logical consistency. And this is an example of that, right? So there is a task that we are operating, which is on ethical inference, so moral reasoning. So these are typically simple situations where you have a statement, such as the one on the top. After asking my daughter first, I discussed my daughter's health problems with other family members, giving vague details and ask for advice. Is this a moral action or an immoral action, and why? And then, yeah, predict with LLMs only simpler prompts. You get this type of explanation and the prediction that this is an immoral action. And if you do that iterative cycle with the symbolic solver, in this case, Isabel, you change that into a moral action, and then you give that argument, which is the fact that it is a moral action, because, yeah, essentially there was a form of consent behind that. And you can see that the quality of the explanation, the articulation of the explanation is more detailed and more consistent, more stepwise, right? So that's what you expect this to go in a logical way, in a more controlled. So to conclude, large language models are indeed a monumental development and game-changing foundation for doing inference. And you can think about this as a kind of efficient, not fully controlled substrate, right, that you can use for language especially, but also for other data modalities, right? Omics emerging as a data modality. It allows this fluidity and lowering this impedance mismatch between different representation elements. So going from a graph database to a natural language statement, relational, right? So this allows code to natural language to code. And this, it's really a very efficient way that you can put all your evidence, all your background knowledge into the same shape. Is practically very important. So, but alone, they're not fit for purpose for scientific reasoning, for clinical reasoning especially. So the question that I try to answer are some emerging architectural patterns, modeling pattern that supports evidence-based reasoning, explanatory reasoning, that could bring this vision of binding this lower barrier to evidence to control the reasoning. And I discussed in particular methods around variational encoders and neurosymbolic architectures, trying to give a couple of examples of that. So thank you for your attention. In case you have any questions, very happy.